Hi, everyone. So when we left off with Chapter 12, Part 1, we were discussing cell division by mitosis. And I just want to point out that cell division by mitosis, that is what happens in eukaryotic cells. Because if you think about what's going on there, you are getting rid of the nuclear envelope, so you're talking about a nucleus, and you're also talking about a cell that has multiple linear chromosomes. So let's switch gears and let's talk about how cell division occurs in prokaryotes. Now, remember that prokaryotes have, of course, no nucleus, right? You guys know that. But also their genetic material is in a bit of a different form. Instead of being in multiple linear chromosomes, prokaryotic cells have one chromosome, and it's circular. Hopefully this rings a bell for you guys. Definitely something I would want you to know, say, on a final exam. So if I were to draw a bacterium, like up here, and I were drawing the genetic material, it would just be a single circular chromosome. Things to note: recall that prokaryotes are much smaller than eukaryotes. Also, they're much simpler. So they have much less genetic material, so less DNA overall. These guys are just simpler. You just don't need as many genes that provide the instructions for how to make a bacterium as you do to provide the instructions to make a human being, right? So prokaryotes, much simpler. Therefore, their process of cell division is also much simpler. Their process of cell division is something called binary fission. Binary meaning two, fission meaning to break apart, right? So binary fission is how we're going to take a bacterium and break it in two. So it's kind of similar, but much faster. The first thing that happens is the DNA is copied, and we have a whole lot less of it to copy because we only have this one chromosome here, and this one chromosome contains so much less DNA, okay? So here we go from one chromosome to, hopefully you can kind of make out that we now have two chromosomes. Another thing that I hope you can make out is we have some kind of a protein string here. So we've got some sort of a cytoskeletal element, a filament, that is attached to each of these chromosomes. So we have one attachment here, and it looks like we have another attachment there. So what now happens is that these protein filaments, these cytoskeletal filaments, they're going to pull these chromosomes apart, as you see there. And at the same time, you can see the beginnings of a protein ring, a cytoskeletal ring that's forming in the middle here. The cytoskeletal ring starts to pinch inward. And so we now have a situation where we have one chromosome and a completely pinched off cell membrane. And we have the same chromosome here and a completely pinched off cell membrane. So much like mitosis, binary fission results in two exact copies of the cell. We're going to have separation of chromosomes as well as cytokinesis. But you can see that it happens much more quickly. We don't have to get rid of the nucleus or separate out a whole bunch of different chromosomes. Much, much simpler process. Now let's take a second and step back and think about eukaryotes again. So we're kind of backtracking now. I want to move back to eukaryotes. And I want to think a little bit about how eukaryotes can control when they divide and when they don't. Okay, so I told you earlier that there are some eukaryotic cells, like, for example, neurological tissue, that as you're developing, your neurons, your neurological tissue divides, your cells divide to make brain tissue and spinal cord tissue. But then as you reach a particular stage of development, those cells stop dividing and they get arrested in the G1 phase. And what happens is, if you get a brain injury, those cells typically don't regenerate. So they spend a long time without dividing. Other cells divide much more rapidly. So what controls how often these cells divide? Well, it turns out that there is a substance called M-phase promoting factor that determines how often cells divide. Now, M-phase promoting factor is actually made up of two components. The first component is a protein called cyclin, and it was named cyclin because researchers noticed that it cycled up and down in its concentration according to when the cell was dividing. The second part of M-phase promoting factor is abbreviated CDK, but that actually stands for cyclin dependent kinase. And that's in your notes down below as well. Now let's take a look up here. So here we have the M-phase promoting factor together. We've got cyclin here, and we've got the cyclin dependent kinase, or CDK, here. Actually, let me erase that because I'm going to want to draw something there. So what happens is, throughout the course of interphase, remember that interphase is a combination of these three phases here, G1, S, and G2. Throughout interphase, the concentration of cyclin goes up in your cell, okay? And then as it reaches a threshold value, okay, here's the concentration of CDK. It's always pretty constant. So when the concentration of cyclin matches the concentration of CDK, and you have a certain concentration of this dimer here, okay, I said dimer. Dimer means a two-part molecule. Once you reach a threshold concentration of this dimer, it triggers the cell to move into mitosis, to move into M phase. And once it does, the concentration of this dimer, or actually of the cyclin, I should say, starts to decrease, okay? Well, let's talk about how that happens. So let me just do some erasing here. So as I noted earlier, cyclin increases throughout the course of interphase while CDK remains constant in terms of how much is in the cell. So the amount of this is going up, the amount of this stays the same. And as the cyclin is produced, it attaches to CDK. But normally what happens as this concentration is going up is that there is a phosphate here, okay? This phosphate is called the inhibitory phosphorylation site. So this phosphate inhibits the action of these two things. What happens is, after the cyclin reaches a certain concentration that is equal to the CDK concentration, there's an enzyme that will come along and it will take off this phosphate group. When it takes off that phosphate group, what it does is it activates, so let's make that up here. The removal of this phosphate is going to activate CDK. Now, I'm hoping that you'll remember from earlier lectures that I told you that any type of protein that is called a kinase, that is an enzyme whose job it is to phosphorylate things. So what does it mean if you're phosphorylating something? You're adding a phosphate group. Typically, phosphorylation turns proteins on. It activates them. Okay, so when the concentration reaches a certain level for this cyclin CDK dimer, there's an enzyme that will come along and it will take off this inhibitory phosphate. And what will happen instead is this thing will activate and it will work to turn on proteins that initiate the M phase here, okay? And it does that through a process of phosphorylation. Now that's how we get this process started, but notice that right after we turn M phase on here, the concentration of cyclin starts to go down again. Why is that? Well, throughout M phase, and in particular in anaphase, there are enzymes that are produced that degrade, I was going to say chew up, but we'll just put degrade, that degrade cyclin. Oops, cyclin. So what that means is this combo turned a bunch of enzymes on. Those enzymes started the process of mitosis. But throughout mitosis, additional enzymes come along and make a product that chews this guy up, causing this concentration to go down again, which is going to shut off the division process, okay? And this will happen again and again throughout the course of the cell cycle. This right here, where enzymes come back and degrade cyclin, this is an example of negative feedback. And if you need to be refreshed on what negative feedback is, let me encourage you to go back to the enzymes chapter, which I believe is chapter 8, to review this concept. Negative feedback is a concept that I believe you should understand before you leave introductory biology. And so I will for sure be asking about that in the future, okay? So this is how we can go through, we build up our cyclin concentrations, and then when it reaches a threshold value, we start mitosis. Then some of the mitotic enzymes degrade cyclin to bring it down to the original level. Then cyclin gets produced again until it reaches a threshold. Then we start mitosis. Mitosis is going to create some enzymes that degrade cyclin to bring us back down, etc. Okay? Now, this cyclin CDK, let's put it here, cyclin CDK, this combo of cyclin and CDK, it turns out that there is more than one version of cyclin, so I'll put many, and there's more than one version of CDK. So this example here that I showed you, it works as a way to start M phase, but it also, using different cyclins and different CDKs, that's exactly how we turn on different phases of interphase, okay? So right here, there's a cyclin CDK combo that turns that on. And right here, there's a cyclin CDK combo that turns mitosis on. And right here is a cyclin CDK combo. Wherever we have cyclins and CDKs turning on phases of the cell cycle, those are called checkpoints, cell cycle checkpoints. And there are 3 cell cycle checkpoints that are operated using different versions of cyclin and CDK. So let's start here with what we call the G1 checkpoint, okay? And I'm just gonna put G1 checkpoint. All right, let's look at what happens in the G1 checkpoint. The G1 checkpoint occurs after the cell has spent some time in G1, and remember that G1 is when the cell is growing and doing its cellular functions, and just before the cell goes into DNA synthesis. there's a few things we want to check out to make sure that the cell is ready to enter DNA synthesis. So if the cell size is big enough, if there's enough nutrients available to the cell, if there's something called social signals that are present. Social signals are often something called growth factors, and we're going to talk more about those in a second. Think of these as hormones that signal that the cell population is at the right size for the cell to undergo division. And then the last thing is there are enzymes that check to make sure the DNA is not damaged. You don't want to copy a bunch of DNA that's damaged, right? And so if the DNA is damaged, then this cell will no longer divide and it will just die. So at the G1 checkpoint, these things have to be satisfied. And if they are, then the cyclin CDK combo is going to, I don't want to do that right now. Then the cyclin CDK combo is going to send this cell into S phase, okay? So that's the G1 checkpoint. There's also a G2 checkpoint. When does that occur? So the G1 checkpoint occurs after G1, right? So I bet you can guess when the G2 checkpoint occurs. It occurs after G2. So here's where our G2 checkpoint is, here, right here where this purple is. And at this point, the cell is checking, have the chromosomes been replicated properly? Is the DNA undamaged? Is there enough MPF? And remember, MPF is this dimer here, but it's the one that sends it into mitosis, okay? If these three things are true, then the cell will enter mitosis and cell division. Then there are two checkpoints in mitosis. So we're going to call them the M phase checkpoints. And I'm going to put plural checkpoints. They occur at the end of mitosis. Okay. And again, these are checking to see, have the chromosomes attached to the spindle apparatus, okay? And have they properly segregated, and has MPF been degraded? So the take-home message here is that all along the way, there are enzymes that check whether or not the cell is damaged or whether the cell is functioning properly. And if it is undamaged and functioning properly, it will send the cell into the next phases. The idea being that you don't want to copy a cell that has some kind of an issue with it. But sometimes we do copy cells that have issues with them, and that is what happens in cancer. Cancer is really a disease of mitosis, because what's happening in cancer is that mitosis does not properly shut off. Cancer is essentially uncontrolled cell division. And what happens is, like in this example, we have, I'm going to say that this is maybe like an intestinal cancer because this looks like sort of an intestinal setup. These are all normal, healthy cells that we have here in pink. These cells in blue are tumor cells. And tumor cells are any rapidly growing cells that take over, and they grow so fast that one of the things you notice about tumor cells is that they are something called undifferentiated. Now, what does that mean? Well, because they are, they undergo undifferentiated, because they undergo, hold on, undifferentiated, there we go. Okay. Because they undergo mitosis so much, they don't have a lot of time to spend in the growth phase where they do their little cellular functions. And so they tend to not have, they don't have the proper, and I'm going to put that in quotes, proper characteristics. What do I mean by that? Well, let's just say that you're a liver cell. There are some characteristics that all liver cells share, right? There are some characteristics that all intestinal cells share. These cells, they're just kind of general cells. They don't really look like intestinal cells. And what that means is, because they're just generalized cells, as this tumor gets bigger and bigger, and some of these cells start to detach they're kind of generalized. They can just kind of go and stick any other part of the body. And when that happens, when cells detach from a tumor and colonize other part of the body, you may have heard this term before, that's when we say that the cancer has metastasized. Metastasized. In other words, the cancer has been able to colonize another part of the body. The reason it's able to do that is because the cells don't really look like any particular cell. They just look like general cells. So if this thing goes and tacks onto a bone cell, the bone's like okie dokie, and it just hangs out. Now, how is it that cancer causes disease? Well, the way that it causes disease, the way that it makes you sick, is because it's a sneaky, sneaky thief. Cancer cells, they rob the neighboring tissues of nutrients, okay? So if you're making a bunch of cells here, right? These cells are feeding off the nutrients that really should be going to healthy tissue. Additionally, these cells disrupt function of your organs. What do I mean by that? Well, let's take a look at our diagram. Here you got some healthy intestine. Here you got some healthy intestine. Presumably, this part of your intestine can absorb nutrients from your food, as can this part. But this whole part right here, this isn't functioning properly. And so now this individual that has this cancer, their ability to absorb nutrition from their food is compromised. So now I've given you sort of a general description of cancer, but it turns out that there are more than 200 different types of cancers in human beings. And they all have a little bit different characteristics in terms of what exactly went wrong in mitosis, where they're growing, how fast they grow. So there's no magic bullet that we can use to treat all cancers, because of these 200 types, they all have individual characteristics. And one scary statistic is that 40% of Americans will develop some kind of cancer during their lifetime. So be vigilant. Whatever screening tests that you are eligible for, especially as you age into populations that need to be screened. For example, I'm in the population that needs to be screened routinely for breast cancer, and I make sure that that happens on an annual basis. So just be aware that the odds are kind of high that you or someone you love will have cancer, and you need to detect it early. So just to kind of continue with this idea that there are 200 different types of cancer and they're very common, I also want to point out that these different types of cancer, because they are have different problems with mitosis, they vary in their lethality. So for example, breast cancer is really common. So you can see here, this blue column is the estimated number of new cases. That's a lot. But look at the number of deaths. That's pretty low, okay? And one of the reasons for that is we have really good detection. We can detect breast cancer really, really early. And so we can do things to try and treat it ASAP. Lung is more deadly. So we have this many new lung cancers, and we have this many deaths that result from lung cancer. Again, people don't typically detect it until there's something wrong with them. And by then, usually they are pretty far along in their disease. So you can kind of work your way through this list to compare how many cases are caught each year versus how many deaths occur each year. All right. Now, one way that we can treat cancer is by simply removing the tumor. And you've probably heard of something called benign versus malignant tumors. So a tumor is just a cellular overgrowth. So the tumor, so I'm going to put cell overgrowth. So the tumor is what results from mitosis not turning off properly. So here we have a tumor, and you can see, look, we've got, looks like this is an anaphase cell. No, this is a telophase cell probably. Here's an anaphase cell. Here's one that's in prophase. And you can see that this tumor, it doesn't belong here. And we have uncontrolled mitosis here. But notice that there's a difference in this tumor here and this tumor here. This one's what's called benign, and this one's what's called malignant. Another way to think of this is malignant tumors are often considered invasive, whereas benign tumors are non-invasive. Okay, so what that means is if you were to go in as a surgeon, you could surgically remove this and scoop it out. And as long as you got all of the tissue, as a matter of fact, surgeons often try to take a little bit of healthy tissue next door to make sure that they got everything. As long as you get all of that and you remove it, then you're typically okay. All right. Malignant tumors are invasive. And the problem with malignant tumors is you can see that it's going to be a lot harder for you, if you were a surgeon, to go in and scoop this out because this thing is spreading. Okay. So there are two basic defects that can cause this unregulated cell growth or these tumors to grow. Okay. Two basic defects. And I have them for you down here in your notes. First of all, there can be defects in that activate proteins required for cell growth when they shouldn't be active. So there can be activation of mitosis proteins when there shouldn't be activation. Okay, that's one problem that can occur to cause this unregulated cell growth. And then the second problem that can occur to cause this unregulated cell growth is that there can be defects in something called tumor suppressor proteins. These tumor suppressor proteins are proteins that are designed to help shut down cell division. So if you leave the proteins on that are involved in mitosis for longer than you should, you can end up with a tumor. Or if there is some sort of a problem with these tumor suppressor proteins that are designed to shut down mitosis, that can also lead to a tumor. Let me show you an example of how that happens. This is an example of what happens in a cancer called retinoblastoma. This is a cancer of your eyeball. And we have this one pretty well worked out, which is why we're going to use it as an example today. Now let's talk about what happens normally on your retina of your eye, okay? Normally, for your retinal cells to divide, what happens is there are growth factors, which are just proteins that stimulate cell division. Usually it's, you know, if you have like enough cells there, they'll secrete enough of this growth factor. And this growth factor acts like a hormone. It does two things. It turns on, it promotes cyclin production. So it turns on cyclin production. So that's going to build up. Remember how cyclin went up, 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 and then it went down during mitosis. And then it went up, 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 and down during mitosis. Okay, so cyclin is going to go up, 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 up in response to these growth factors. There's a second thing that gets produced in response to these growth factors, and in this example is something called E2F. E2F is called a transcription factor, and I'm just going to put trans factor here. But what it really does is it turns genes on, okay? So turns all right. Now, normally what happens to this E2F is that it immediately gets bound by this protein here. This protein here, RB, is a tumor suppression protein, okay? And the reason it's called a tumor suppressor is because by binding up this thing, by binding up E2F, what this tumor suppressor is doing is it's making this unable to turn on genes for cell division. Okay, so this tumor suppressor is blocking this guy from turning on cell division genes. Okay, now again, what would happen in a normal scenario is that eventually you get enough cyclin to build up and it's bound to this kinase. This cyclin kinase dimer comes along and it gets activated. When it gets activated, then this kinase phosphorylates our tumor suppressor. The tumor suppressor lets go of this guy and he moves on and turns on the genes necessary to start cell division. But sometimes there's problems. One of two things can occur. First of all, some individuals have a faulty version of this, okay? What would happen if you had a faulty version of that? Let's think that through. If you have a faulty tumor suppressor gene, okay, and you don't have this guy here, then the minute these growth factors come along, this can bypass all these steps and go straight to turning on the genes for cell division. That can result in a tumor. Okay. What's the second problem that can occur? Well, the second thing that could happen, the second way this could go wrong, I said, was it that we're going to have the proteins turned on for mitosis when they shouldn't be turned on. So in that scenario, what happens is this overstimulates cyclin production. So instead of producing the appropriate amount, it produces way, way more, which causes this thing to happen earlier, which means that you have more cell division, okay? This is sometimes called altered social control, and that's because these growth factors are considered social control. So when we see changes that result from growth factors, sometimes this is called altered social control. But the big thing that I want you to remember is that cancers can result from these two things: activation of mitosis proteins when they should not be on, or a problem with tumor suppressor proteins. In our example here, if you had a tumor suppressor protein problem, again, just one more time, if this wasn't working properly, then this would bypass the whole system and start the process. If we have a situation where these things are overproduced, this gets turned on before it should, and the thing goes faster than it should, okay? So that's everything from chapter 12. Remember that you will have a quiz. If you're watching this in the fall of 2020, you will have a quiz on October hold on, let me check my calendar. I believe Friday's October the 9th. Let me just double check. Yes, on October the 9th, you will have your quiz and it'll be on chapters 12 and 13. As always, let me know if you have any questions. Talk to you soon. Bye-bye.